This is a podcast of the Nuffield Department of Medicine. Today, Dr. Brian Marsden tells us about his research, from information to structure. Hi, Brian. Can you tell us a little bit about your research interests?
I work for an organisation called the Rockefeller Genomics Consortium, or SGC, which is interested in trying to solve the structures of human proteins and making them available in the public. And one of the key things that we're particularly interested in is understanding how these protein structures inform or create disease.
Okay.
And as part of the work associated with that, we generate a lot of data. So my research is actually interested in how we capture that data and be able to ask questions to better understand how these mutations, for example, in proteins may cause disease.
How do you use an informatics approach to support the work done at the Rockefeller Genomics Consortium?
We have a very multidisciplinary team at the SGC. We have people who are doing cloning of genes, for example, creating proteins, solving the structures of proteins, even people who are doing medicinal chemistry, making small molecules to interrogate those proteins. So one of the things that we have to do is to be able to capture this information in a way that makes sense and is easy to mine. And so as a consequence of that at the SGC, we're entirely paperless. And what that means is that everything that we do is captured in electronic format. And this means that we have access to information from 10 years or more, no matter whether people are still with us or not. And one of the other really important aspects of being able to have everything paperless or electronic is that we have the ability to ask questions or spot trends in the work that we're doing, finding out what we're doing right, what we're doing wrong, and spotting things that we would not have seen otherwise.
How can visualization tools help with drug discovery?
At the SGC, we solve many protein structures of human proteins. And these make very pretty pictures, but they're not particularly accessible to those people who are not structural biologists. And so it's really important that we find ways to make this information available to those who don't understand how to interpret these structures. So something we pioneered at the SGC over the last 10 years is the ability to be able to show these protein structures in the context that makes sense to the scientists. So for example, we can show protein structures in a web browser directly connected with a narrative in an interactive form. And what this means is that people like medicinal chemists, for example, can now look at these protein structures and see and understand the context of what that actually means and help them to understand how they can make decisions about what sort of compounds they may make, for example.
What are the most important lines of research that have emerged in the field over the last, say, 5 to 10 years?
When we started at the SGC over 10 years ago, the sequencing of the human genome was a really big thing, and we really expected that would be a big game changer in terms of what we do. I would say that the revolution hasn't happened as quickly as we would have expected, but these days it's possible to sequence a human genome within a day or so for less than $1,000. So that means you can now ask questions like, what exactly is a single cell doing on its own in an organism? And so we can now understand the interplay between how cells in the pancreas, for example, or in the brain may be working with respect to each other. And no two single cells are the same. So this means that what those cells are doing may be very different. We can also ask bigger questions. For example, we can do something called metagenomics. And this is the study where we look at environmental effects that may be occurring in a bacterial colony. So a classic example may be the bacterial colony that you have in your gut. And it's been found that the sorts of bacteria you have or the flora that you have in the gut may actually influence whether you have things like irritable bowel disease, for example. So by sequencing all these bacteria, we can have a better understanding how that interplay may occur with disease and so on and so forth.
Why does this line of work matter? Why should we fund it?
Drug discovery is an incredibly expensive thing to do, and there are no guarantees that a drug that you think you've got is actually going to work out. Something we want to do at the SGC is to find ways to find the diamonds in the rough, effectively. What are the right targets? What are the right genes? What are the right proteins that we should be working on to actually get new medicines quicker, more cheaply, and in a more relevant way?
How does your work fit within translational medicine within the department?
Recently at the SGC, we've been awarded a large strategic award by the Wellcome Trust, which is focused on us developing something called target enabling packages. These build upon the work we've done at the SGC for many years, so protein structures, how to make proteins, what small molecules we should use, but packaging them up together into something which people within academia and industry can go away and use to better understand whether the proteins associated with those packages are really the real deal in terms of drug discovery. And what we hope is that therefore this will accelerate the translational aspect of the work that we're doing.
That's great. Thank you, Brian. That was really interesting.
Thank you.
Thank you.